So on behalf of Arubadi Video Institute of Technology, Vinaya Missions Group of Institutions, I welcome you for the International Webinar Day 3. Today's topic is role of omics in recent trends in plant biology. Ma'am, is it audible, ma'am?
Yes, sir. Yes, sir.
Okay.
Please go ahead, sir.
Slightly dripping down. You can go ahead, sir.
Okay. So now I request our beloved HOD to give the welcome address.
Thank you, Madhu, sir. Good
Thank
evening
you.
to one and all. It's my great pleasure in welcoming you all for the third day of our international webinar series on trending technology. It's a very great honor for me to welcome a very senior most and eminent educationalist, Dr. Madhavan, Assistant Director, Department of Biochemistry, University of Nebraska, USA. Sir, on behalf of our management, campus director, principal, vice principals, heads of all the department, and on my own behalf, I welcome you, sir, for this session. I am indeed glad to welcome our dynamic Vice Principal, Academics, Dr. S.P. Sangeeta, who is always a supporting and a guiding personality behind our all the departmental activities. I take this opportunity to welcome our Dean, CSIR, Giri Rangasamy, evergreen, energetic personality behind in all the departmental endeavors. Welcome you, ma'am. Last but not the least, I welcome all the budding scientists, biotechnologists who are all attending this webinar continuously and supporting us throughout active deliberation. And I welcome you, all of you. Thank you.
Thank you, ma'am. Now I request our young Dr. S.P. Sangeeta, ma'am, Vice Principal Academics, to deliver special address. Welcome, ma'am.
Thank you, Dr. Madhusudanan. Respected chief guest of today's webinar, Dr. Madhavan Soundarajan, professor from University of Nebraska, Lincoln, USA. Beloved Jemima Madhavan, ma'am. Our beloved Dean, ma'am. HOD, Dr. Devika, ma'am. Biotech faculty, colleagues, and my dear students. On very good evening to one and all present here. I'm happy to address during the international webinar series on trending technologies in biotechnology. On behalf of management and on my own behalf, first let me thank Dr. Madhavan for accepting our invitation to deliver a lecture on the role of omics in recent trends in plant biology, which is a very interesting topic, of course. Though I am not a biotechnologist, out of my own interest, I searched in the website and what I've tried to find what is it. And with the great demand in global food supply, omics technologies would definitely help us in meeting the challenges faced in plant biotechnology. There is no doubt for all of us, sir, that hearing to an expert like you is going to be a big treat for all of us. I congratulate our Dean, Madam, and Dr. Devika, HOD of Biotechnology, for their great efforts in organizing this series of webinar for this whole week. ABIT is striving hard to impart quality education even during these lockdown days through webinars like this. I request everyone to get benefited out of this. Thank you all.
Thank you, ma'am. Thank you very much for your special address. Now I request our dynamic Dean, Dr. Giri Rangasamy, Dean CSR VMRF, to give a special address. Please, ma'am.
Thank you, Dr. Sangeeta. You may not belong to Department of Biotechnology, but you are very close. Though your department is civil engineering, Devika often tells me you both do a lot of coordinated work. Thank you for the nice words.
Thank you, ma'am.
Okay. A special, of course, we thank Dr. Madhavan, and a very special thanks to Jemima, Mrs. Madhavan, who worked with me in Women's Christian College for a number of years. And then I'm very happy that our friendship is still continuing. And she used to visit Chennai often. Then we used to have a lot of good times. Thank you, Jamie. You have taken some time to see what to hear what your Veetikarar is saying, as well as how we conduct this program. I'm very happy that Devika suddenly coordinated, and within a week she could fix all these persons. Devika, I would like to thank you also. It's a very good effort. Now, all of you know that today we are celebrating the International Doctors Day. So I take this opportunity along with you to salute the noble profession that is medical field, the doctors. And at this time of coronavirus and its grip of the whole world, we should recognize the services rendered by the doctors. So all of us, on behalf of the university and our college, doctors around the world, we salute thee and we appreciate all your good work. Okay, coming to our international webinar program. Today is the third day. I thought the first day I can speak. I don't know what made Devika think that every day I have to speak. So today is the third day, and yesterday I was telling something about biotechnology. Today my concentration will be on the miraculous technology internet. I thought I'll start with the 2-3 quotations. If you use technology right, it could give you power and privacy, says Dr. Corey. Technology brings the excitement, helps look into the future, and makes us brave enough to try shaping the world. Elbert Hubbard rightly says, one machine can do the work of 50 ordinary men, but but no machine can do the work of one extraordinary man. So today we have an extraordinary man, Dr. Madhavan, who has been working in the US for a long time. This is very true when you give it a thought. That is, no machine can do the work of an extraordinary man. Man's creativity and innovative power with his brain is unimaginable. We are stuck with technology when what we really want is just a few stuff that works, says Douglas Adams. Coming to our webinar focus on trending technologies. I can add that technology development may draw upon many fields of engineering, of knowledge, including scientific engineering, mathematics, linguistics, and historic, to achieve some practical result. Technology is often a consequence of science and engineering, although it is a conscious human activity. Now, I can just say that supposing we take the renewable energy technology or waste recycling technology. So my PhD is from the environment biotechnology where I studied the water quality. There I have seen that we have cereal ponds which are called the oxidation pond. So you have to prepare the pond and collect the dirty water which has been coming from the washrooms and other places, and then inoculate them with the algae which are highly photosynthetic. So human effort is also necessary, and now all the industries have to produce certificates saying they have primary, secondary, or tertiary level of treatment. So the technology will be meaningful and fruitful because of the hard work put forth by the people. Technology development brings in economic growth, no doubt. It also enhances wealth on the one hand by increasing the income levels of the country on the other. Technology development makes very important contributions. The economic, social, and cultural development. It is constantly developing, and with its exponential growth, there is much to look forward in its role. That is the role of technology in putting an end to world's poverty. So yesterday I was telling the 4 capital P's, which the technologists say that we have found solutions. And one of them is poverty. I've told poverty, population, protein malnutrition, and pollution. So today I thought I'll just say a few words about poverty and the role of technology in that, especially after the invention of the technology, which is miraculous. It's a miracle, and that is the internet. According to Harvard University, technology can be a driving force to lift families out of poverty. Enhancing access to information and communication with the internet facilities can be a boon to several people who reside in villages. The knowledge provided through the net can maintain health, educate families, and open the doors for the poor and unfortunate individuals for schooling and even for higher studies. Loans from banks are helping families in rural communities to come out of poverty. When women have the capability to start up their business, they have the opportunities to invest their money in other areas in their lives. All this is through the internet and the mobile phone facilities. Loan facilities sustain development in poor countries and expand economic growth. Those simple and sophisticated, through simple and sophisticated techniques, the internet can help eradicate poverty. They can even educate people, sustain the environment, and create healthier population. People in developing countries also have access to this technology. Business people invest now with mobile technology, mobile banking services also try to help people. I would like to conclude. Technology is the golden ticket to achieving the goal of ending poverty by 2030. Thank you.
Thank you, ma'am. Thank you very much for your delightful special address by explaining the methodological importance of the technology, ma'am. Now, I request my colleague, Dr. R. Balachandar, to give the introduction about the today's speaker.
Was the Department of Biochemistry, University of Nebraska, participation. The topic is PhD important from
Bala sir.
There is some problem.
Sir, audio. It's not clear. Bala sir. Bala sir.
Shall I proceed, ma'am?
Yeah, you can carry on, sir. Please, sir.
Good evening to everyone. It gives immense pleasure to introduce today's chief guest, Dr. S. Madhavan, Professor, Department of Biochemistry, University of Nebraska, Lincoln, USA, on this occasion. I welcome you, sir. He completed his PhD in botany, specialization in plant biochemistry from Brigham Young University, USA. He has 6 years of postdoctoral research experience from 1981 to 83 and 1986 to 89 as postdoctoral research fellow in Department of Biochemistry and Biophysics, Texas A&M University, College Station, USA. He has totally 38 years of teaching and research experience, out of which he has 23 years of teaching experience in University of Nebraska, USA, and 15 years of teaching experience as professor in Madras Christian College, Chennai, Tamil Nadu. His area of research in carbon and nitrogen isotope fractionation in plants, lignin biosynthesis in switchgrass, carbon sequestration in agri-ecosystems, role of acetylcholine in signaling process in plants, carotene and natural rubber biosynthesis. He has been received various prestigious awards in 2019 College Distinguished Teaching Award, and in 2013 he has chosen as a honorary fellow in the Innocent Society from University of Nebraska, Lincoln, and many other prestigious award he has received. And he is also an active member in various professional societies like American Society of Plant Physiologists, American Society for the Advancement of Science, International Society of Photosynthesis Research, National Collegiate Honors Council, and Asia Pacific Chemical, Biological, and Environmental Engineering Society. He gave various invited presentations in national, international seminars and conferences. Today, he will enlightening us with topic role of omics in recent trends in plant biology. On behalf of Department of Biotechnology, once again, I welcome you, sir.
Thank you.
Thank you, sir.
Am I ready to go?
Yes, sir.
Yes, please,
sir.
sir. Please, sir.
Okay. Once again, thanks for inviting me to be a participant in this international webinar series. Let me first congratulate the organizing committee for all these things. And my special thanks are due to Dr. Madhusudanan, Dr. Devika, the head of the department, Dr. Sangeeta, and of course, Dr. Giri Rangasamy, whom we know for eons, for past many years. So what I wanted to talk to you today about, and all the listeners who are listening and the participants who are participating in this webinar series, I thought I'll talk to you about some of the recent developments in omics that has caused the advancement in plant bioscience. And to the extent the knowledge that we have gained in plant science is due to the development in the omics technology. I am going to be getting out of my comfort zone here, because if I were to give a talk, usually I would prefer to talk on natural rubber or keratin biosynthesis or biofuel or any of those things. But then I thought for the budding scientists, it will be a good idea for me to talk about the techniques that are available to advance this cause and understand how one can utilize it in the maximal capacity to unravel the mysteries of science, especially in the plant science world. Can we go to the slides, please? Do we have the presentation with you?
Sam?
Sir.
Yes.
Oh, thank you.
Yes.
Yes, it is projected.
Oh, good.
Mm.
Okay, got it. So I thought I'll talk about the role of omics. When I say omics, it includes a variety of technologies. So the group term omics includes a lot of things, and I will try to give a brief understanding of each one of those techniques in the next few minutes, and how it has helped, these different techniques have helped the plant biology, advances in plant biology. So in order for us to understand this, one has to understand what is plant metabolism is, how exactly the plant metabolism can be unraveled through the advances in knowledge in technology. Many years ago, when we studied plant metabolism, we would talk about photosynthesis, respiration, nitrogen nutrition, growth and development, and so on and so forth. But then in a very general background, now because of the advances that we have with this omic technology, we are able to integrate all the knowledge that we have gained through the understanding of the DNA technology, the protein machinery, and the small molecules that play an intricate role in understanding all these things. So this is an integrated study, integrated understanding of how exactly the whole system works, constituting the so-called term system biology. So when we talk about plant metabolism, it lies on an intricate network of genes, proteins, and metabolites. We all understand what genes are. We know what proteins are. And if we have to group metabolites under this, with these macromolecules, that makes it very significant because metabolites are usually considered as small molecules. How can these small molecules play a big role or a very significant role in regulating plant metabolism? So in order for us to understand this, I'm going to give you a very brief intro to plant metabolism. So, and also one has to figure out how plants are able to change or adapt themselves to the changes in environment. So if plants are human beings, they can shield themselves from cold, heat, other kinds of stresses by taking a shelter. Plants do not have the luxury of doing it. They have to withstand the heat, the cold, the stresses, both water and minerals, and so on and so forth. So in order for them to respond to this, they have to adapt or develop some adaptive mechanisms. And how these adaptive mechanisms are going to be regulated is mind-boggling. And that's what they do. And in order for us to understand this, we need new tools and approaches. And that's our, I thought I'll talk to you a little bit about those new tools and approaches grouped under the collective term omics. So metabolism in plants is not a haphazard process. It is not uncontrolled. It is always regulated. So how exactly is this regulated? And how do we know that it is regulated? And what kinds of regulatory mechanisms are present in these plants is the one that I'm going to say a few words, and then we'll go into the omics thing. So metabolic regulation in plants. Why is this important? We all know plants photosynthesize. When they do photosynthesis, they make carbon compounds. And when they make carbon compounds, they provide skeletons for the other synthesis to occur. So when they make these carbon skeletons, we can incorporate a nitrogen to make them into a proteinaceous compound. And then we need to have a balance of these protein-rich as well as carbon-rich compounds to support the optimal growth of plants. And integrating into the system is the nitrogen, the phosphate, the other minerals. So all these things have to be regulated. The levels of all these things have to be regulated in such a way that these plants can respond to their alterations in the environmental conditions. So let's think very briefly about some of the metabolic things that we are all familiar about. For example, if you take every living organism does glycolysis, every living organism does citric acid cycle. So if you put these two, I mean, I'm not going to go into the details because that'll be boring, but with just these two different cycles that are present in every single living organism that we know of, these two have 18 reactions. And these 18 reactions are mediated by 18 enzymes. And to manufacture these 18 enzymes, plants and other living organisms have to invest so much of energy. So that energy is utilized to make those compounds. The same energy is released when these compounds are broken. So there is a balance. They are always at an equilibrium. So that equilibrium is maintained to adapt themselves to these varying changes in the conditions that they have to face in the environment. So the systems have put in place several regulatory mechanisms. So if, for example, if you take glycolysis, for example, if you take glucose and then convert that into the next reaction, which is called glucose 6-phosphate, the plants and the other living organisms always maintain that homeostasis, their equilibrium in shape. In order, no compound is going to be maximally expressed at the expense of another compound. So everything has to be placed, everything has to be at a minimal level, so they'll have an optimal machinery to work with. So what I'm trying to say here is regulation is the key, whether it is one reaction or hundreds of reactions. Now, I just gave 18 reactions for just 2 cycles. There are many, many, many reactions occurring in a cell, and they're all compartmentalized. So think about this. This is a very well orchestrated, choreographed condition. It is like a musical symphony. Every single organelle in a cell has to participate. Every single organelle has to be regulated, and collectively they bring the cell to focus. They make the cell to function. And thousands of cells have to coordinately express themselves, form a tissue, and then the whole organism. So think about that. Think about that for a minute, and that should give you goosebumps as to how these cells can do all these things without having to go through any external. The only thing that they receive is the external stimulus. And I'll talk about the signal transduction in a few minutes. So how does these are regulated? How all these mechanisms, how all these conditions are regulated? As I said before, the plant that you see on the thing is Arabidopsis, a close relative of mustard plant. And when you grow this plant, it can be subjected to the changes in light conditions, heat, cold, drought, salinity, heavy metals, so on and so forth. And how do they then maintain these things? So one of the things that they do is to express the proteins, and to express the protein, the DNA has to be activated. So the DNA has to make RNA, and here on the RNA has to make the protein. So in order for the proteins to express, you need to have transcription factors. These are called transcription factors. So these transcription factors are very selected, and they are selected to express only few particular proteins. So when they see the secondary stresses like osmotic stress or oxidative stress, you have the equilibrium changed, damaged, disrupted. So they make certain functional proteins. And then these functional proteins, going through different mechanisms, through kinases and other G proteins, produce the so-called secondary messengers. This is the place where, when I talked about the small molecule, these are the secondary messengers that play a significant role in elucidating the rest of the scheme. So the rest of the pathway are dependent on these secondary messengers, and these secondary messengers are the ones that regulate the transcription factors. And then you have gene activation to be regulated, to control. So collectively speaking, every single structure that you have in a living cell, whether it is a plant, animal, microbial, it doesn't matter. Everything has to be activated, everything has to be regulated. And that's the reason why this slide is there. And from an organismal level to the cellular level, you have the same kind of regulation. You have the mechanism for regulation for metabolism. You have the epigenetic regulation, what it really means is outside of the normal genetics, epigenetics regulation, and the transcriptional regulation. Most of you probably know transcription is from the information transcribed from the DNA to the RNA. So that's the transcriptional regulation that is regulated. And then you have the translation, and it can be either a transcription or post-translation regulation. So everything is regulated. Everything is under equilibrium. So this is one example where a leaf senescence, leaf senescence is expressed. So if you look at the diagram, I don't want to go into the details here, but if you look at this, the most impressive picture there is the chloroplast with all its components there, with the grana, the stroma, the lamellae, and so on and so forth. So one can see when a leaf senesces, the organelle structure of the chloroplast is disrupted. So what happens to it? Is it going to go as a waste, or the cell has a mechanism to recycle its components? Where did we get this idea of recycling? It is through the cell. Nothing is going to go to waste there in the cell. one of the means by which the recycling occurs, especially with reference to proteins, is to first make the proteins to make them function, catalyze different enzymatic reactions, and make the products and so on and so forth. When once the function is over, then these proteins have to be broken apart. So the individual components of the protein can be utilized for the next synthesis. This is what is called as proteolysis. Lysis means breaking down. So breaking down of proteins is important for regulating the metabolism. One of the organelle that plays a significant role is the vacuole. Vacuole is the place where proteases are generated, and these proteases are then diverted and utilized for lysing the various proteins that the different organelles have. And then they can be reutilized. So not only do we have these signals that we have inside, but also when the pests and pathogens activate the plant defense, they also provide different signals. So once again, the organism has to employ different mechanisms or strategies to defend themselves from these pathogens. So they have to understand or make some or engineer themselves into certain compounds. So like ABA, the abscisic acid, or ethylene deficient mutants, these are the case studies that we have done. And also the jasmanic acid or the salicylic acid and so on and so forth. So it is the intricate intermingling of these different compounds that help these plants to regulate, to defend the effects of pests and pathogens. So not only do they have to maintain a normal life, but they also have to maintain or defend themselves from attacking pathogens. So it adds another layer of complication during their lifetime. So they have to develop certain strategies, and that's what they do all the time. It is mind-boggling, and that's what every single organism, whether it is a plant or animal or microbe, it does. And this is a lesson to be learned. One of the most important necessities for a living organism when they make the proteins is to modify them to become amenable for the particular function that they are destined to function. So every single protein, all proteins cannot do all functions. Every single one is very specific to the role. So how does the living organism know how to characterize each one of them, or how to qualify each one of them to carry out that particular function? So some proteins, when they are made, they can be phosphorylated by adding a phosphate group to a single amino acid, or they can be attached to a lipid, so that becomes a different protein. They can be attached to small molecules like ubiquitin, or they can be attached, they can form disulfide bonds. So, or they can have an acetyl group, CH3 group, attached to the N-terminus of a protein, so the N-terminus of the carboxy terminus. So if you look at all these things, just to give you an example, these are not the only way by which they are the post-translational modifications occur, but just to give you a glimpse of the techniques that these organisms employ to modify the proteins to suit the needs of the reaction that they have to carry out. If that does not give us an idea as to how complicated this system is, yet they don't show their complexity by just performing their function. There are various signals that the plants have to negotiate. Now be patient with me because I haven't gone into the omics. All these things that I'm talking about, all these, the knowledge that we have gained is because of those techniques. So when the plant receives the signals to defend, to alter its physiology, to alter its mechanism, because of the various signals, as I said, the gravity, the temperature, the light, the wind, the herbicides, the pathogens, The root system, you have the nutrients, the water availability, the soil structure. Collectively, so much of a stress is laid upon a plant when it grows. And they have to adapt. The phenotype that we see is very much related to the genotypic function. This is what one has to understand and appreciate, the significance of each one of these signals that the plant receives and adapt themselves to develop certain strategies. How do they do it? I'm not going to go once again into the total biochemistry of this because that's a little bit more complicated, but generally speaking, if there is a signal, that signal, if it is a compound that will bind with a protein that is on the membrane. So the plant membrane play a significant role. So the plant membranes are made up of lipids and proteins. Some of these proteins can be a receptive compound molecule so that the signal can be attached to this receptor protein, which in turn would turn on the proteins or transduce the signal in the cytosol through various pathways. So through kinases or G protein coupled complexes or whatever it is, that signal is perceived, whether it is external or internal, that is perceived. And then that triggers a cascade of pathways. And that's how the cells regulate themselves, the cells, and then it carries on to the organismal level. Once again, this is a complicated picture, but I'll just give you the ligand attaching to the lipid or to the protein, turning on the G protein complexes, and then which would turn on the cascade of pathways, resulting in the production of secondary molecules, which are very effective in regulating certain things. So the take-home message from this slide is the signal is perceived, the signal is transduced, and that relates to, that affects the stimulation of the pathways and production of secondary molecules. Okay, so as I said before, I wouldn't be able to talk anything about the things that I talked about before in the earlier slides if we had no knowledge of the omics and currently the bioinformatics. What do I mean by omics? And what do I mean by bioinformatics? The related terms for all these things would be computational biology or systems biology. So when we talk about bioinformatics, this is a very integrated approach. So you collect massive data from various sources. You store those data, and then whenever it is necessary, you retrieve it, you organize it, and you analyze it. So the analysis of the biological data to generate useful biological knowledge constitute the so-called bioinformatics science. This is a very upcoming developing field. One has to have some computational biology experience. You need to have some computational experience, mathematics experience, but just basic things will be able to help developing the bioinformatics knowledge. So when we have all this knowledge, not only can we study what has happened, but we also can forecast what is going to happen to an extent. To an extent, that is the underlying statement. We can certainly analyze these things to the nth degree and come up with certain strategies, suggestions for combating some of the effects that the living systems have to face. So what are these omics? Genomics. Genomics, as the name suggests, when it has to do something with the gene. So if you are talking about the gene, then you are immediately thinking about the nucleic acids like the DNA, RNA, and then the individual constituents of all these things, the nucleotides. So you talk about the nucleotide sequences, and then if you take just one genome, hundreds and hundreds and hundreds of genes. So you sequence everything, you find the particular gene, you annotate the gene, and then you align the sequence, and then see whether that sequence is homologous or has any homology to other organisms. And then you analyze them comparatively, find out the phylogenetic relationship, and then see whether there is any mutation and make a functional prediction, and so on and so forth. So from this, you would have sort of understood the intricacies involved in collecting this data and utilizing to the fullest extent to understand the relationship that exists between organisms, and then find out where exactly the mutations have occurred. Very relevant for this COVID-19. So it is not one RNA virus. We have several of these viruses. So where do they differ? And how do they differ? Where exactly do they differ? What nucleotide has changed from one thing to the other? We can predict all these things, and we can come up with some solutions. Taking it to the next level, from genomics to the transcriptomics, this is where the RNA expression levels are counted. So the DNA transcribes the information to RNA. That's why this is called as transcriptomics. So you have the RNA expression levels, and then you generate the transcription factors. So how exactly these transcription factors, which are nothing but proteins, but the binding of these transcription factors regulate the RNA expression, and then find out the chromatin structure and so on and so forth. Then you do the same thing, like clustering, functional enrichment, and the regulation, and so on and so forth. And then the next level of this omics is the proteomics. So from the DNA to RNA, where we have expressed, so the DNA sequence, now we know what particular gene has to be expressed. We have it in the messenger RNA, and then we are going to translate that into protein. So proteomics comes into vogue. So where we now understand, we can find out what the protein levels are, because we know the expression levels of these things. Once we have, we can selectively make a particular protein. We can understand its structure, and we can understand the different interactions that the protein has to have. And then we can identify the protein. We can once again make a functional prediction, structural predictions through X-ray crystallography and other techniques that we now know of. And then we can simulate this through molecular dynamic software. And then we have, once again, we can talk about the mutation. In this particular instance, it's going to be a single amino acid substitution. For example, if you know of the hemophiliac disease from the normal red blood cells, just one particular amino acid from on the sixth place in this particular one, if that is in the hemoglobin, if it is changed, it becomes hemophilic. So from one change in the amino acid in the sixth position can make it a hemophilic blood cell than the normal cell. We know because of all these techniques. We came to know about all these things because of our ability to sequence them, to understand what has changed, and so on and so forth. Now, this is a big one. And then, of course, as I said before, small molecules also play a significant role. So metabolomics. So we can analyze the metabolites. We can understand how these metabolites, the pathway network analysis, and then they regulate the whole system. And then, of course, we have the imaging and cytometry. And all these things constitute the so-called systems biology. This is the jargon of the age. This is everybody talks about systems biology. Systems biology is nothing but a whole system approach to understand all these things. So it encompasses everything that we have talked about so far. Okay, so if you look at the, don't look at the right side panel of the slide, but left side panel gives you an idea as what the bioinformatics really mean. Going from the bottom to the top, from the genome to the transcriptome to the proteome to the metabolome, and then finally to the phenome. So if you look at this one, from a molecular level to the cell level, to the small molecule level, and then to the organismal level. That's what the bioinformatics, the whole concept of bioinformatics gives us, an understanding of the complex nature of all these things put together to understand a simple structure. One of the things that has helped us in utilizing all these omics is the genetic engineering of chloroplast genome. This is not my study, but I'm going to borrow the information from one of our best friends, Dr. Henry Daniel, who is at the University of Pennsylvania. He has been a pioneer in engineering chloroplasts to modify its function to produce variety of compounds that are useful for human growth and development, and so on and so forth. So the whole picture that is given here is how exactly he went about doing all these things. It's a complicated slide, but the take-home message here is he could engineer the chloroplast to provide the functionalities that are necessary to produce compounds such as antigens for several compounds, like for some of the vaccine antigens like dengue fever, polio, malaria, tuberculosis, cholera, anthrax, and plague, and also some autoantigens, diabetes, hemophilia, and so on and so forth. All that he has done is take the chloroplast as a system or a vector, introduce all these things, modify the chloroplast genome to the extent to produce these compounds. It is not a simple task because this is a plant cell, so it has its own deficiencies. But because chloroplast has its own DNA, this is semi-autonomic structure, so he can engineer that one to make these compounds produced in that system. And recently he has produced some lettuce cells, freeze-dried lettuce cells, and after expressing some of these biopharmaceuticals, and they can be stored for about 30 months at room temperature, which is a remarkable feature. And then he can also do several things like blood clotting factors, extending angiotensin, and so many other things. So all in all, this is a remarkable study. This would not have been possible if we had no idea of what these omics have done. The next level is on the phloem road. So most of us are interested in how plants flower, why do they flower at a particular time? And this is, of course, related to the photoperiod. Most of us know this. But then if you go into the molecular level, this is because of the signals that they have. So there are transcription factors like CO or the florigen like FT protein. This is the florigen transport protein, so it is in the phloem. So all these things are possible. We know now of this existence of all these things because of the omics technology, because we are able to analyze them through a variety of techniques. And I cannot make a presentation without making a reference to biofuel, because I've done some work on this one for a long time, especially on switchgrass, which has cellulosic biofuel, which is different from the grain biofuel and the algal biofuel. But the cellulosic biofuel, the most important component is the cellulose, and the ability for us to break the cellulose. We, human beings, with all our knowledge, cannot break cellulose. We don't have the enzyme to do it. So we are dependent on the bacterial systems to produce cellulase, and they are the ones that are utilized for breaking the cellulose from the fibers of switchgrass and so on and so forth to produce the bioethanol. Once again, as I said before, this is what I did for a few years. So biofuel produced through the biomass of switchgrass is the most sought after. Even the corn biomass now being utilized more than the corn grain. Omics have played a significant role in developing phytomedicines and phytochemicals. I'm not going to talk about this. I'm sure you have heard more about this from Dr. Ramya Mohanraj from Houston. But most of the institutions in Chennai and other parts of Tamil Nadu have actively, they're doing a lot of studies, research in this area. And I've gone through several theses on this one. So, but since you have already heard from it, I'm not going to talk more about this. But once again, the significance of omics research has given us a new knowledge on the phytomedicines and phytochemicals. We also know that plants have neurotransmitters, neuroregulators, and neurotoxins. One should ask the question, why do plants have neurotransmitters? When we talk about neurotransmitter, we immediately come to think of the animals and the human species. One of the most important neurotransmitter that we have is acetylcholine. Acetylcholine is a compound that is in the synapse. So it has to bind and give us the electrical signal so we can have the functionality. I mean, our coordination, our thinking, everything is based on the transmission of information from cell to cell. And that cell to cell communication is through some of these neurotransmitters like acetylcholine, like DOPA, like so many other things. So in the mid-90s, we were interested in studying some of these neurotransmitters' existence in plants, and we found the presence of acetylcholine. And then we immediately asked ourselves the question, why does it exist in plants? Of what use does this compound have in regulating the plant's life? There are many neurotransmitters like melatonin, hyperforin, excitotoxin. The hyperforin most probably, is an antidepressant. Excitotoxin is the substance that they add to the foods and beverages, like in monosodium glutamate, the MSG, and the NutraSweet, and so on and so forth. So all these things are available in plants. So as I said before, we did some work on the acetylcholine, especially the enzyme that breaks down acetylcholine. We published this, and the take-home message here is plants have the acetylcholine machinery, same as the human beings, but they have a different function. They have a cell-to-cell communication function, but they have nothing to do with the neurological issue, but in ion transport and so on and so forth. The very last one or two slides is on the omics also has given us an understanding of the ancient flower. If you look at a flower and if you look at the structure, we only are mesmerized by the beauty of it, but also the arrangements of it. And in order to get that arrangement, in order to find out how the genes have given those things, one has to understand the different genes that constitute all these things. So one of the ancient flowers, and then the knowledge that we have gained, it's depicted in the right-hand panel. You can see there's so many, we have a huge wealth of information assembled together to give us an idea as to how a single flower can be developed. Very last one is the phenome and phenotyping. This is where the whole organism is taken into account to understand. And here at the University of Nebraska, we have both a field phenotyping facility as well as the greenhouse facility. What is the advantage if you are going to be asking that question? What is the advantage of having this? The advantage is not only can you study a single plant, but you can study multiple rows of plants at the same time using the facilities that we have in camera and so on and so forth. So the very last slide that I'm going to show you is the Greenhouse Innovation Complex that we have at the University of Nebraska. We have an automated high throughput plant phenotyping platform. So we can, each group of plants can be stressed or can be subjected to a variety of conditions. and we can study all of them at the same time to get a global understanding of what these things do. So summarizing all these things, it is not too much to say that the knowledge that we have gained through the employment of different omics technology has given us a hope to advance this science further and further. Thank you all.
Thank you, sir. Sir, actually our UG students are started isolating the phytochemicals from different plants, and they are going with the nanoparticles also, synthesis of nanoparticles. So
Extremely.
it's going on,
Yeah.
sir. So we are taking it up and we are all publishing also, sir. So this is the information from our side in our department, and it's going on.
Yeah, I didn't stress too much on that one because I wanted to give a holistic talk.
Yes, sir. Yes, sir. Thank you, sir. Thank you. It was a highly informative one. Dr. Madhu, sir?
Thank you, ma'am. Thank you, sir. Thank you very much, Professor Dr. Madhavan Soundarajan, sir, for sharing excellent, enlightening, and entertaining session on omics, the right way to apply with other areas. He has explained from genetic engineering of chloroplast, biofuel, switchgrass, phytomedicine, phytochemicals, neurotransmitter, neuroregulators, and neurotoxin, and ends with application of these. So I hope that the participants also enjoyed the show. Now, there are a few questions from participant side, sir.
Sure.
Mr. Justin Anthony has asked, is the word omics refers to gene, proteins, and metabolics?
Say it again.
Is the word omics refers to gene, proteins, and metabolics?
Yeah, omics is a very general term. It can be genomics for genes, it can be proteomics for protein, it can be metabolomics for small molecules, it can be lipidomics for lipid-associated compounds, it can be glycomics for carbohydrate. So there are several. So each group, as they develop, they add the term omics to make it more significant.
Thank you, sir. Next, Shashikant has asked, will an amino acid in a protein undergoing post-translation modification can undergo mutation too?
Yeah, well, one example that I gave is the hemophilic cell, where the 6th amino acid is changed to valine, which is what to say, very which is different from the normal amino acid. So just by displacing or substituting one amino acid, one can change the entire architecture of this particular hemoglobin. It is just one change, and this is due to mutation. And so, yes, it can be done. And this is what most of the people, most of us, I have done in the past, but not right now. But one can do what is called a site-specific mutagenesis. So we can take out one particular amino acid from a protein, insert another protein, insert another amino acid, and see what behavioral change that protein displays when once this change has occurred. Yes, it is site-specific mutagenesis.
Thank you, sir. Then Dr. Pauline Orr is asking, when do plants lose the race in the wild, especially to changes trigger, especially to challenges triggered by human activities and fail to adapt themselves?
I didn't get the question correctly.
When do plants lose the race in the wild, especially to challenges triggered by human activities and fail to adapt themselves?
It's a very tough question to answer. Yes, human activities cause a lot of damage. If the activity is for developing a system to magnify that particular organism, it is all for good. But if it is for countering the existence of the plants, and its ability to combat itself through this various things, it is very difficult. So that's a very complicated question, and one has to be well, I am a narrow biochemist, so I don't know how to answer that question.
Thank you, sir. Thank you very much, sir. Mr. Selvi Gnanasegaran has asked, can we say that adaptation and regulation are quickly achieved by microbes rather than higher plants, animals? If so, are they more evolved?
Very good question. Yeah, very good question. So this is the symbiotic association with the microbes is very well known between plants and microbial systems. So this is the, I'm sure Dr. Rangasamy will also understand, or rather give an idea about these things in nitrogen metabolism, in nitrogen fixation. So you need to have a symbiotic association with the bacteria in the root system, which is beneficial for both the plant and for the microbes. So whether that is going to have an impact on an evolutionary trend is a question that an evolutionist will answer. But there are other examples like Klebsiella that not only we have association in the leaf areas, but also in other systems. So if one can stretch imagination and say, yes, these adaptive strategies employed by both the microorganism and the plant are there to enhance their evolutionary trends.
Thank you, sir. And Mr. G. Varadaraju is asking, sir, I have gene sequence. Is there a way to go with genomic studies? Lack of financial facility, I did not do genome sequencing. Please give ideas to connect my work towards omics.
Well, if you have done genomics, you have already done a small aspect of genomics. Gene sequence, if you have done gene sequencing, you have already done small part of genomics, because that's where the whole thing begins. Gene sequencing is a way to understand the different nucleotides. And if you have done the gene sequencing, and if you find another sequence of the same gene in a different organism, and if you find some small changes, then the ability for you to understand and compare the sequences is a part and parcel of genomics. So yes, you have already started the process. There are more complications because you know from your molecular biology understanding, not only that particular nucleic acid can be changed because of mutation or other things, or it can be some purine change or pyrimidine change. It can be dimer forming and so many things. So we don't know whether what is the source of that change, what brought that change, and then you collect more data about these things and then analyze it. And when you collect the data, you organize, you will have a dense assemblage of data constituting the so-called metadata. And then you go through and then you enter into the field of bioinformatics. Yes, you are starting the system. So that's extremely good.
Thank you, sir. Thank you very much for explaining.
Can I ask a question?
Yes, please.
Yeah.
Yeah. From the papers and the television shows, we come to understand that COVID-19, as it entered the world, now is going a number of mutational changes. That is the spike protein. Is it true?
Yeah, coronavirus has a lot of tentacles. So, which means it is going to produce different proteins. And so which protein is the one that is going to be attached to the cell? And that is the target now. I think most of the universities are developing the vaccine for. So is this the protein that one has to go after? We don't know the answer to it until the vaccine comes out, and whether the efficacy or the efficiency of that vaccine is going to tell us as to whether this particular protein is the one that one has to go after or not. But as I said, the very pertinent question in terms of what kinds of protein that this organism produces, we have knowledge. When I say we, that is a rhetorical we. People have knowledge of the kinds of proteins that are expressed by these coronaviruses. We do know that. But which particular protein that one has to go after is the one, because there are mutations to these ones, and one has to study the mutated forms of this protein, because coronaviruses have been in existence for many, many years. Yeah. This particular coronavirus is a mutated version that we have seen. So that's the reason why this is causing so much distress. Did I answer your question, Giri?
Yeah, yeah.
Thank you, ma'am. Thank you, sir. So one more last question. Justin Anthony has asked, how genetic engineering related to computational biology?
Oh, genetic engineering, well, let's put it this way, computational biology, we did genetic engineering even before the term computational biology came into existence or came into vogue. So genetic engineering has been done for many, many years. And that paved the way for collecting so much of data. And when everybody had their own way of doing their genetic engineering, and when we assembled all this data together, then the computational people came and then said, well, we can put this data together and analyze this data through creating some algorithms and then software development. That's how the bioinformatics or computational biology came into vogue. And so if one has to do it the other way, it would have been nice, because computational biology would have given us some understanding as to which direction genetic engineering has to go. But we did the other way around, which is also good, because then we are now in a position to develop so many tools to understand this complexity that we never would have found otherwise.
Thank you, sir. Then the same person has asked, please throw some light on system biology and how it have its implications in future.
System biology is a collective term. As I said before, when you understand the genomics, the proteomics, the metabolomics, the lipidomics, the carbohydromics, whatever you want to call it, glycomics, when you collect each one of these segments, each one of them can provide you with a lot of data. So when you put them all together to understand one particular system, one particular organism, that constitute the so-called system biology. So the implications of those things in the biological understanding of a single system, the so-called system biology, it is a mind-boggling study, because if you understand one system, then you have to know everything about it. And I don't think one has a lifetime to understand everything, so we segment them into separate units.
Thank you, sir. And Mr. Vasudevan is asking, you were mentioning about switchgrass for biofuel through the use of cellulase. What was the biogas volume? Is it comparable to biogas production from vegetable waste?
Oh, absolutely. Vegetable waste also has cellulose, mostly cellulose. If you have utilizable non-starch compounds, or starch compounds. Starch compounds can be broken through amylases and other enzymes, so we can produce them into small molecules. But celluloses that are present in the cell walls, we cannot break them until we employ an enzyme. And we don't have the enzyme as human beings. That's why we take fibers. And we don't know how to, we don't have the capacity to digest it. So we need the microbial systems to produce cellulase to break those small, to break down this polymer. if you compare this cellulose with starch, both are made up of glucose units, nothing. They are the same, exactly the same. Both cellulose and the starch polymer are the same, except for the binding, the bonds that they have. In starch, you have the alpha 1,4 bonding, whereas in cellulose, we have the beta 1,4. We don't have the enzyme to digest cellulose because we don't have an enzyme to break that beta 1,4 bonding. But we have for the alpha 1,4. The amylases can break this. See, the small change makes us not able to digest cellulose. So nature has so much complexity. Why it has developed this particular enzyme to produce to digest alpha 1,4 and not beta 1,4 is mind-boggling. But that is what we have. So it is definitely an advantage for if we can use the non-utilizable celluloses to be converted into some utilizable form in ethanol by employing cellulases which are available in some microbial system. So that's the difference between the vegetable waste with the biomass waste. Other biomass are going to go. I mean, plants produce a lot of biomass, so we can utilize them to the nth degree.
Thank you, sir. Thank you very much.
Somebody wants your email ID, Dr. Sridevi. So can you type it now? Devika.
I'll pass it to Ma.
Yeah,
Dr. Sridevi wants his email ID.
Yeah. Yes, ma'am. I'll do it.
You can give it to her.
Yes.
Thank you, ma'am. Now I request Dr. A. Nirmala to give vote of thanks.
Thank you, sir. Good evening, one and all present here. It's my privilege to propose a vote of thanks on this occasion. My sincere thanks to Principal, Vice Principal, Dean, and heads of the various departments for their support and guidance. I would like to express my special thanks to our chief guest of the day, Dr. Madhavan Sundarajan, Professor, Department of Biochemistry, University of Nebraska, USA, and also my special thanks to Jemima Madhavan for gracing today's webinar and delivering a wonderful information in role of omics in recent trends in plant biology. Thank you so much, sir, for your very interesting and thought-provoking address. I am sure that all the participants have taken a note of your suggestion and information. It will be very useful for their future. I express my deep gratitude to our beloved Dean, Dr. Giri Rangasamy, for her vision, interest, good leadership capacity has been another success story of this 5-day international webinar. Thank you, ma'am. I also extend my special thanks to our HOD, Dr. R. Devika, for her enormous cooperation in the organization of this event. Thank you, ma'am. I am happy to express my special thanks to all faculty members in the department for their support in different ways. My special thanks to person who have been a backbone for the function, Mr. Sam, for making all the arrangement for this webinar. My special thanks to all the participants who have been turned up from various places in Tamil Nadu and other districts. Without you, the program will not be a successful one. We thank you so much for your great cooperation. Thank you, one and all. Thank you, ma'am. Thank you, sir.
Thank you.
Thank you, ma'am.
Someone wants the PPT also. Madhavan,
Yes, ma'am. Yes, ma'am.
You have
Yes,
it.
we have. Let them, you can, dear participants, you can just meet and ask for the PPT. We'll share you.
And Dr. Selvi is there. Selvi?
She's muted.
Mute.
Yes, ma'am, I'm here.
Good evening, ma'am.
Good evening.
Evening. You can say a few words, Selvi.
I was not fortunate to be. I was not fortunate to be taught by Dr. Madhavan, but then I have heard him in so many places. And today was yet another session where I really enjoyed his talk on regulatory mechanisms, because my PhD work was on regulatory mechanism in cyanobacteria. And that made this talk much more meaningful to me. Thank you very much, sir.
You're welcome. You're welcome. Thanks.
Jamie ma'am. Hello. Hello, ma'am. Nice seeing you. I was her student.
Oh, okay.
In
Ma'am?
UCC.
Jemmy ma'am?
Jimmy,
Not to be seen.
can you hear? Jimmy?
She has to unmute.
Mute
Yeah,
பண்ணிருக்காங்க.
yeah.
Madhavan, thanks a lot, Madhavan. It was very
Oh,
interesting.
you're welcome.
And
Sir, thank you very much, sir.
You're welcome. Thank you again for giving me this opportunity.
Yes, sir. We are actually fortunate to have you today, sir. All of us.
and I think twice he has visited AVIT. Okay, we'll wind up, Devika.
Yes, ma'am. Thank you very much, ma'am. Thank you.
Okay. Thanks.
Thank you very much.
Thank you.
Sir.
Thank you, sir.
Thank you, ma'am.
sir. Take care.
Have a good rest of the night.
Thank you.
Yeah,
Bye-bye.
Bye. Bye.
Bye.
Bye. Bye.
Bye.
Bye-bye.
Bye.